proteins that are being actively being synthesized by that host cells, right? For example, you have a parasite, you have a toxin, you have even Rickettsia intracellular microorganisms, you have Mycobacterium tuberculosis, you have Leishmania, Legionella, you have a bunch of that kind of. They always use our cells or our ribosomes in order to produce their own proteins. They cannot do it by themselves, so they require the host cells to make it for them, right or not? Even when you have a cancer cells, the proteins are made inside that cell. That cell is not normal, right? If you have a cell that has been invaded by a specific type of virus, that cell is not normal no more, right or not? Become abnormal. So that's what's happening here. So proteins in the cytoplasma of that cells, right, are being degraded into peptide by certain proteasomes, and then they will be transported into the endoplasmic reticulum by the type 1 or type 2 some proteínas transportadoras, right? But remember, what they do is they carry the peptide to the cell surface and will present it to the outside of the surface so that the cytotoxic T cells can encounter them. So the cytotoxic T cells that will go there will recognize that peptide bound to class 1. Remember when the cytotoxic T cells were in the thymus, the way how they will keep the CD8 was because they could recognize an antigen sufficient, well, an antigen that was presented by MAC1, right? That was positive selection, don't ever forget that. And failure to positive selection was when that T cells, I don't mean CD8 or CD4, when that T cells, for example, couldn't recognize the antigen presented by MAC1, we said, well, that's a blind T cells, then we need to kill it, right? Remove it. And then the other was when that T cells recognize the antigen presented by MAC1 too much, with high affinity. Could cause you autoimmune disease. We said that's what we call negative selection, right? And require what? The process of apoptosis will be ignite inside that cells, and that will happen. So that's what happening here. So they can recognize that's antigens, and that's the signal that triggered the release of that cytotoxic molecule. We said the perforin, that will result in the destruction of that abnormal cells, right? Remember, class 1 can recognize peptide that come from the host cells, all right? Because when you go into MAC2, they recognize peptide, non-endogenous peptide. These are called endogenous peptide because they are being produced from within. The other is an exogenous peptide. For some reasons, the phagosomes will phagocyte the cell, will process that into smaller peptide, and then MAC2 will present it into the cell. That was MAC2. Remember that, right? Exogenous, this is endogenous peptide. It's being produced by that cell. That's why when you have Mycobacterium, that's why when you have virus, they cannot replicate by themselves. They require the host cells to produce some specific protein for them. Yes or not? That's when we know it's abnormal. This is the same picture. So associate type 1, type 2, endogenous peptide, MAC1, cytotoxic T cell, abnormal cells. And then we go for the class 2. We said that you have an alpha and a beta change, and we have alpha 1 and alpha 2. Remember we said that, right? And beta 2, the function of class 2 is to present, looking here, exogenous peptide. All right, exogenous means a foreign peptide, all right, non-self. The other were endogenous because it was produced by that cells. Caramba, si usted tiene un cáncer cell, ¿dónde se está produciendo? Right. El otro viene de afuera y se metió para adentro, nomás que la célula lo fagocitó, el antigen presenting cell. Lo procesó y dijo, Vamos para afuera, man. ¿Qué es lo que va a ocurrir generalmente con el T-helper 1 y T-helper 2 que estábamos diciendo? Pues? ¿Se acuerdan ya el mecanismo? El antigen presented by macrophages will release interleukin 12 y ahí va a activar la conversión de la T-helper into T-helper 1, right or not? Pero no sabemos el tipo de célula que fagocita al otro tipo. All right. They are found only on B lymphocytes, macrophages, monocytes, dendritic, son todos sistemas fagocíticos. Right? Activated endothelial cells and activated T cells. Dijimos eso. Yes or not? All right. Aquí está el peptide binding group que habíamos dicho. Comparando con ellos, aquí está. The protein from outside, exogenous peptide, le decimos, are internalized by the cells, right? Are being phagocytized by the cell, and you can see this. It's like an endosomes in here, all right? Usually, once they get inside, certain endosomes will fragment those, will process the protein into smaller peptide, all right? Don't forget the invariant change too, all right? The other important thing is right here, as you can see, we have the alpha and beta, we form the MAC2, and then we have the invariant change, will protect the peptide group, right, from being occupied and also from proteases. Will move to these backwards until they will join with the previous endosomes in here, and they will form a larger one. Both of them, that's the point where the invariant change will be degraded or released, and they will encounter those smaller peptide. Remember, that was a larger one, and then they will process that before. Once they present that one, they will take it to the surface, and that's what we said. The MAC class 2 peptide complex then transported to the cell surface when it's going to be recognized by helper T cells, which have antigen receptor that only can recognize antigen presented by MAC2. And that antigen presented by MAC2 is an exogenous peptide, like I told you here. All right? The protein comes from outside. All right? Okay? The other comes from inside the cell. Don't forget that you have an alpha 1, alpha 2. Remember the other one you also have, but you have alpha 1, alpha 2, alpha 3, and beta 2. There's no beta 1. And the peptide group is occupied by the number 1. Remember alpha 1 and beta 1. Remember that one. All right, and the other was an alpha 1, close to the amino terminal. Here, amino terminal, here, the carbon. Both are transmembrane. In MAC1, only the alpha 1 was transmembrane. The other one, aquí está. Okay. Ok, yo creo que estaba todo eso. Y luego están algunos ejemplos. Dijimos entre el class 1 y el class 2, these are all nucleated cells. These are only macrophages, B lymphocytes, activated T cells or endothelial cells will participate in here. MAC1, 1 times 8, 2 times 4. Remember? Recognize endogenous peptide, exogenous peptide, right? That will be processed later on. They will act as a target cells, as the cell to be eliminated, and the other will present to helper, and then helper will continue what happened with the page 62 that I told you, right? Okay. Class 1 contain an alpha, beta 2 contain alpha and beta. All right. Those are remember, this is a light change and a heavy light change, heavy change. All right. Okay.